Welcome to Goldfish Evo Devo.
Sounds fun.
Yeah, you might be thinking goldfish. Really? What's so special about, a pet store staple? But honestly, this seemingly simple creature offers a surprisingly deep dive into evolutionary developmental biology.
It really does.
And the star today really is the goldfish's amazing twin tail.
Ah, yes, the twin tail.
It's more than just a pretty feature, isn't it? It's like a keyhole view into this fascinating interplay between evolution and development.
Precisely. And to really get into that, we're diving into a specific study. It's quite recent research from Kinya Gyota, Gembu Ab, and their colleagues.
Okay.
And what they did was closely examine the muscle structure of these twin tails. They compared it to regular single-tailed goldfish and also to zebrafish, which are a common model organism in biology.
Okay, so comparing the fancy twin tails to the sort of standard fish tails, what were they hoping to find by looking so closely at the muscles?
Well, the big question, really, is how did this very unusual twin tail evolve? Specifically at the muscle level. What actually changed in the tissues to create this, double structure?
Right, how did it physically happen?
And maybe more importantly, what can this tell us about the bigger picture of evolution? Like, how can existing developmental toolkits, the genes and processes organisms already have, be adapted or reused to produce totally new traits?
Okay, let's zoom in on that twin tail then, because, if you put one next to a regular fish, it's really striking.
Oh, it truly is. A classic example of what biologists call divergent morphology. It's a major departure from the single sort of continuous tail fin that most vertebrates have.
And this didn't just appear randomly in a pond somewhere. This was intentional, right?
Absolutely. This goes way back. Since the Ming Dynasty, so centuries ago, goldfish breeders have been deliberately selecting fish with this trait, generation after generation, purely for how it looked.
Wow, okay. So purely for aesthetics. Anatomically, though, what actually makes it a twin tail? What's going on under the surface?
So the research confirms what breeders probably knew implicitly. There's a duplication of the caudal skeleton. Those are the bones right at the very end of the spine. And also the muscles that control that skeleton. So basically where a single-tailed fish has, one tail unit, a twin-tailed goldfish effectively has two.
A whole extra set of tail bits. That seems like a pretty fundamental change. Is that common in fish?
Well, mutations causing twin tails can pop up occasionally in other fish species, sure.
Yeah.
But the key thing with ornamental goldfish is that it's become a stable, inherited trait. It's genetically fixed, specifically because of all that selective breeding.
Ah, right. It's part of the breed standard for many fancy goldfish.
Exactly.
Okay, so we have this very deliberately cultivated, quite unusual trait. What does its existence tell us about how these fish develop from an egg?
That's where it gets really interesting for Evo Devo. The fact that the twin tail could emerge and become fixed suggests that this artificial selection, the choices breeders made, must have somehow altered the underlying developmental mechanisms.
The instructions for building a tail.
Precisely. The ones that normally dictate how tail muscles and bones form. And these are usually really fundamental processes, very conserved across vertebrate evolution. So the goldfish twin tail represents a significant tweak, basically driven by human taste.
So the standard developmental playbook got edited, let's say. And the study actually points to a specific gene involved here, right? Something called cordon.
Yes, that's a crucial piece of the puzzle. A specific mutation in the cordon gene, the allele, is called GHDS127X, is known to be responsible for the twin tail phenotype.
And how does that gene mutation lead to a split tail?
Well, this genetic change happens really early in the embryo. It disrupts the normal top-bottom signaling, a process called ventralization. And this altered early development ultimately causes the tail fin folds, which normally fuse into one, to bifurcate to split into two.
Wow. So one gene change early on has this massive downstream effect, leading to a completely different tail structure. So the researchers knew about this genetic link, but their study went deeper, didn't it? They looked at the actual muscle tissue and found something unexpected.
They did. They found what they called a twin-tail goldfish-specific muscle.
Whoa, okay, a specific muscle.
Yeah, located right in the space that forms between the left and right halves of that duplicated tail skeleton. They've named it the medial caudal muscle.
Medial meaning in the middle.
Exactly. And this specific muscle in that specific location hadn't really been described before in other teleost fish. That's the group that includes most bony fish we know.
Okay, that sounds like a pretty big deal. A potentially brand new muscle unique to these fancy goldfish. So the obvious question is, where did it come from?
Right. Is it a completely novel evolutionary invention? Or is it maybe, a modified version of a muscle that their single-tailed ancestors already had, just repurposed?
Like, is it built differently or just positioned differently?
Exactly. The researchers asked, is this medial caudal muscle totally new in its makeup, or could its structure and composition, if you look really closely at the tissue level, be comparable to muscles found in regular goldfish?
Yeah.
Even if the overall location is new, maybe the building blocks are familiar.
Okay, which leads us naturally to muscle fiber types. You hear about fast twitch and slow twitch in humans. What's the deal with fish? Fast versus slow muscle?
Yeah, it's a similar idea. You can think of it like this. Fast muscle fibers are for quick, powerful bursts of speed, like escaping a predator or lunging for food.
Got it. Sprinting muscles.
Right. And slow muscle fibers are more for endurance, sustained, steady swimming, just cruising along. They're more efficient for that kind of activity.
Makes sense. And is there a typical arrangement of these in fish?
Generally, yes. In the main part of the body, the mid-trunk, most teleost fish have a pretty conserved pattern. The fast muscle is typically found more medially, towards the core of the fish.
Okay.
While the slow muscle is usually located more laterally, on the sides, often just under the skin.
So power in the middle, endurance on the outside? Seems logical for swimming.
Pretty much. Though there are exceptions, like in really highly active swimmers like tuna, where things get modified. But for many fish, that's the basic layout in the trunk.
But I have a feeling you're going to tell me the tail is different.
You guessed it. That was the key finding in this study. When they looked closely at the caudal region, the tail itself of the wild-type single-tailed goldfish, things were more complex than that simple mid-trunk pattern.
How so?
Well, the superficial layers, the ones near the surface, often follow the expected pattern. But when they looked at the deeper muscles in the tail, like one called the flexor caudalis ventralis, they found slow muscle fibers mixed in. And sometimes these slow fibers were actually located quite medially, close to the axial skeleton, the backbone.
Huh. So slow muscle deeper inside the tail, near the bone, not just on the outer edges. That's unexpected based on the trunk pattern.
It is. And what's really interesting is that they observed similar patterns, slow muscle fibers in those deep caudal muscles in zebrafish, too.
Ah, okay. So it wasn't just a weird goldfish thing.
Exactly. It suggests that this more complex arrangement, with slow muscle fibers deep in the tail, might actually be a conserved characteristic in these types of fish. It's not just a quirk of the goldfish, fancy or otherwise.
Okay, that's a really important baseline. Wild-type goldfish and zebrafish already have this more intricate muscle fiber layout in their tails. So now, back to the twin-tail goldfish. What did the researchers see when they looked inside that brand new medial caudal muscle? What was it made of?
Right. So they examined two popular twin-tail varieties, the Ryukin and the Oranda. And they found that this medial caudal muscle contained both slow and fast muscle fibers. It wasn't exclusively one type.
A mix, okay. But was there any kind of organization to that mix? Or was it just random?
No, definitely not random. And this is a crucial observation. They found that the slow muscle fibers within that medial caudal muscle showed a distinct bias that they tended to be located more laterally within that muscle, often right up against the duplicated caudal skeleton on either side. They confirmed this by looking at horizontal sections through the tail, too.
So even this novel muscle has a structured arrangement of its components, and the slow fibers seem drawn towards the skeleton. That is fascinating.
It really is.
Now, the researchers didn't stop with just the standard ornamental breeds, did they? They also brought in some lab strains.
Correct. They took advantage of lab strains of goldfish that had different tail fin shapes, all resulting from various mutations in that same cordon gene we talked about earlier.
Why look at those?
Well, by looking at a range of tail shapes linked to the same gene, they could explore the connection between the specific cordon mutation, the resulting tail morphology, and the muscle fiber arrangement inside. See if the patterns held up.
And what kind of tails did these lab mutants have?
They observed quite a spectrum. Some looked like a normal single tail, others had a narrow twin shape, some a wide twin shape, and some even had tails that were visibly twisted.
A whole gallery of cordon-induced tails. Did these mutants also have that medial caudal muscle? And what were their fibers like?
Yes, several of these mutant strains did show slow muscle fibers within their medial caudal muscles. And importantly, just like in the Ryukins and Orandas, these slow fibers were often situated close to the caudal skeleton.
Okay, so that reinforces the pattern. Medial muscle forms, slow fibers tend to hug the nearby bone.
It does seem to strengthen that link. But there was an exception.
Ah, always an interesting exception. Tell me about it.
One particular strain, designated 24twin#01, showed a different pattern. Instead of that consistent lateral bias for the slow fibers within the medial muscle, in this fish, the slow muscle fibers were just sort of scattered randomly throughout the medial caudal muscle. No obvious pattern.
Any idea why?
Well, interestingly, the researchers noted that this specific lesh also had a rather irregular, kind of disorganized arrangement of its caudal skeleton.
Okay, so let's pull this together. We have the twin tail, the new medial muscle inside it, the mix of fast and slow fibers, and this tendency for slow fibers to be near the skeleton, except when the skeleton itself is disorganized. What are the main hypotheses the researchers are considering for how this specific muscle arrangement evolved?
They're basically weighing two main possibilities, two evolutionary scenarios.
Okay.
The first possibility focuses on selective pressures. Could this specific arrangement, the slow fibers positioned laterally near the bone, actually provide some functional advantage for the twin tail shape that breeders like?
You mean maybe it helps them swim better with those two lobes? Or maybe it just makes the tail look a certain way that breeders preferred?
Exactly. Perhaps it helps control the movement of the bifurcated fin more effectively, or contributes to the aesthetic appeal. If so, breeders might have unknowingly selected for fish with this particular muscle fiber distribution over centuries.
That makes a certain kind of intuitive sense, function influencing form, driven by selection.
It does. But there are a couple of wrinkles that make the researchers hesitate on this being the sole explanation.
Like what?
Well, for one thing, why would a narrow twin phenotype, which might have arisen relatively recently from a single-tail lineage in their lab strains, almost immediately show this specific bias pattern? It seems quick for selection to act so precisely.
Good point.
And then there's that outlier, 24twin#01 with the random muscle distribution. Its existence proves that you can have a twin tail without this precise lateral bias of slow fibers. So it's maybe not strictly essential for the twin tail form itself.
Right. Those points do complicate the simple selection story. So what's the alternative hypothesis?
The second possibility, and the one the researchers seem to lean towards, involves conserved developmental mechanisms.
Okay, back to those pre-existing instructions.
Exactly. They propose that the biased distribution of slow muscle fibers seen in the medial caudal muscle might actually be a consequence of developmental programs that were already operating in the deep caudal muscles of wild-type goldfish and zebrafish. Remember how those deep muscles also had slow fibers near the skeleton?
Yes, I do.
So the idea is, when the twin tail mutation happened, it created this new anatomical space, the region where the medial caudal muscle forms. And the existing developmental instructions, the ones that pattern those deep muscles, might have simply been redeployed or allowed to manifest in this new context.
Ah, okay. So it's not that a new program evolved specifically for the medial muscle, but rather an old program found a new place to run when that space opened up.
That's the essence of it. The evolution of the twin tail provided a new stage, if you will, and the existing developmental actors just performed their roles there.
That feels, I don't know, quite elegant. Evolution as a tinkerer, reusing parts in new ways.
It's a very common theme in Evo-Devo, yes.
Yeah.
Repurposing existing tools.
Now, you mentioned developmental signals. We know the notochord, that rod-like structure running down the back in embryos, is super important for guiding muscle development. How does that fit into the twin tail story? Because even with two tail skeletons, they still only have one notochord, right?
That's absolutely correct. Despite the duplicated caudal skeleton and musculature, the notochord, which is a major signaling center, remains single. And that raises a really interesting question.
Yeah, how does one signaling source pattern development in both the original single-tail context and this new twin-tail context, especially that bit in the middle?
Right. How does a single conductor lead what seem like slightly different developmental orchestras in the tail?
So what are the possibilities there?
The researchers outlined two main hypotheses. First, maybe the development of this new medial caudal muscle is simply governed by the very same signals emanating from the notochord that pattern the main trunk muscles. Those signals are known to influence muscle fiber type determination.
That seems like the simplest explanation, maybe. The existing signal just floods into the new area.
It would be simpler, yes. However, the researchers suggest a second hypothesis might be more promising.
Okay.
The second idea is that the development of the medial caudal muscle might involve different or perhaps additional molecular signals. And crucially, these signals might be coming from the adjacent caudal skeleton itself.
Ah, so local signaling from the bone.
Potentially. The observation that the slow muscle fibers in the twin tails are so consistently found right next to the skeletal elements lends support to this idea. Perhaps the developing bone provides specific local cues that influence the differentiation of the muscle fibers right beside it.
So not just the main notochord signal from afar, but very localized instructions from the immediate neighborhood, from the bone itself.
Exactly. It suggests that while the notochord sets up the broad body plan, these fine-tuned features in novel structures might rely heavily on local interactions between tissues.
That's a fascinating possibility. Okay, so stepping back, what are the really big takeaways here? How does studying the intricacies of a goldfish tail help us understand the grander scheme of evolution, how new things arise in nature?
I think this study is a beautiful illustration of how seemingly dramatic new physical traits, novel morphologies can actually come about. The goldfish twin tail is a perfect example of what they call a novel medial morphology, something new appearing in the space created between duplicated structures.
Like if you had two arms side by side and something new grew in the space between them.
Kind of like that, yes. And what this research strongly suggests is that forming these new features doesn't necessarily require inventing entirely new genetic programs out of thin air. Instead, evolution can be incredibly resourceful. It often reuses and modifies the developmental systems that are already in place, deploying them in these new anatomical contexts, these novel spaces. The specific way the muscle fibers are arranged in that medial caudal muscle seems to be a textbook example of this principle.
So evolution isn't always about brand new inventions, but often about clever repurposing and modification of existing blueprints.
Precisely. And the researchers draw parallels to other major evolutionary events that involve duplication and the emergence of new structures in between. Think about the evolution of paired fins in fish, or even paired nostrils in vertebrates.
Wow, okay. Those are major evolutionary steps.
Indeed, and it's plausible that similar principles adapting existing developmental machinery within newly created spaces between duplicated parts were at play during those transitions too.
It's really quite amazing to think that the humble goldfish, specifically its fancy tail, can offer these potentially profound insights into how major features of animal bodies might have evolved.
Absolutely. The appearance of this new muscle, the medial caudal muscle, with its specific internal organization, really highlights just how flexible and adaptable these underlying developmental pathways can be.
It really reinforces the idea that new forms, new morphologies don't always need entirely new instructions. They can emerge through modifications, through the redeployment of existing processes, and may be guided by very local cues, like the presence of those duplicated bones.
That seems to be the key message. It's a combination of genetic change, opening up possibilities, and existing developmental systems filling those possibilities in structured ways.
It definitely makes you think, doesn't it? When you look at other strange or unique traits in nature, that weird horn on a beetle or an unusual feather pattern on a bird, could similar things be going on?
It's a great question to ponder.
Could these features also have deep evolutionary roots in conserved developmental processes that have just been tweaked or reused in a novel context? It certainly gives you a new lens to look at biodiversity.
It really does. It encourages us to look past just the surface appearance and appreciate the intricate, resourceful dance between evolution and development that has shaped life all around us, even in something as familiar as a goldfish bowl.